I will talk about adjuvants for some time. The word has originated from Latin. Adjuvare means to help, and these adjuvants are used for immunization. For any vaccination, all the vaccines that are prepared till date in the world, they are administered with adjuvant. The reason being, the vaccine that you prepare could be a soluble antigen. And being a soluble antigen, when you inject into the body, that soluble antigen may get lost without being processed by the antigen presenting cells. It may be excreted from your body in a large extent, and it may fail to stimulate an immune response. In order to boost the immune response and make this antigen or the vaccine that is injected to be available for a long duration in the body to the immune cells, adjuvants are used. The adjuvants were discovered by a scientist called as Freud's. N is silent. He's a French scientist. Therefore, it is called as Freud's. Freuds. N is silent. There are two types of Freud's adjuvants. One is Freud's complete adjuvant. What is this complete adjuvant? It is water in oil emulsion, where the oil phase will be paraffin oil, not any other oil, not vegetable oil. It is a paraffin oil. Paraffin oil is also called as mineral oil. It is obtained during the distillation process of the petroleum refinery. And this oil is non-reactive, and it doesn't cause any kind of allergic or any other adverse reactions in the body. And this paraffin oil is the oil phase, and the aqueous phase will be there, and the suspension of bacteria, of tuberculosis bacteria, that is killed bacteria, will be there in the oil phase. The tuberculosis bacteria is a strong antigen. No immune system can be evaded by the tuberculosis bacteria. Therefore, the dead preparation of tuberculosis bacteria is suspended in the oil phase, and it does contain a significant amount of peptide on its surface called as muramyl peptide. Muramyl peptide. Muramic acid you might have heard about. It is a fatty acid, muramic acid. And that muramic acid is conjugated with the membrane protein to form muramyl peptide. It is a lipoprotein of strong antigenic ability. Therefore, it is used as a stimulant. So in the oil phase, the tubercle bacteria, killed bacteria will be there. And then what you have to do is that vial that is available as Freud's complete adjuvant to you, into which you have to inject the vaccine that you have prepared. And generally, the vaccines that you prepare will be in aqueous phase. Occasionally or rarely, they will be in the lipid phase, okay? Or they will be lipid soluble components. Most of them, vaccines, are soluble in aqueous phase. So when you inject that into the vial, that is complete adjuvant vial, the vaccine gets suspended in the aqueous phase. Now you have two phases in the vial. You mix this vial so violently in such a way that it forms an emulsion of milky suspension. Oil and water phase is mixed with a very vigorous shaking that it forms a milky suspension. It will be very thick milky suspension. And that emulsive drops that are formed, emulsion drops that are formed, will trap your antigen, that is your vaccine, in the oil in water phase in such a way that it will be evenly distributed. Now that thick milky suspension is injected into the person to immunize the person, and one can choose the intramuscular. The adjuvant vaccines are not administered through intravenous route. It is very dangerous. It will cause rupture of capillaries because it is a very thick emulsion. It should not be administered through intravenous route. You can administer that as subcutaneous, intradermal, or it could be intramuscular. Okay, but oral vaccines or nasal vaccines or intravenous vaccines are generally or with only aqueous phase, and there will be no oil phase. Okay, there will be no oil phase, and these vaccines need to be administered repeatedly to stimulate the immune response. But now coming to fruits complete adjuvant, the vaccine when you administer in a particular place, most of you might have experienced it, not when you are very small kids, but later if you are vaccinated. If you are vaccinated on your arm, upper arm, that arm will become very painful. In that region it becomes very painful, and for about a week you will not be able to lift that arm. And suppose if you are vaccinated on your buttocks, for 1 week you will not be able to sit on that buttock. It will be so painful because that vaccine is very thick suspension. It forms an inflammation in that area. It contains tuberculosis bacteria and it deposits in that area and gets dispersed very slowly by the blood to different parts of the body and to the lymph nodes through the lymphatic channels. And that antigen that is suspended in the face is going to get distributed in small pockets. In that way, the adjuvant serves to distribute the antigen very slowly in the body and make it available for a long duration for the immune cells, that is especially the antigen presenting cells, to process that antigen which is suspended in the oil phase now in the form of emulsion, and then the immune boosting takes place. And then what is Freud's incomplete adjuvant? It is slightly different from the previous one. It will also contain the paraffin oil and the aqueous phase, but in the oil phase the dead tubercle bacteria will not be there. Therefore, Freud's incomplete adjuvant will be less viscous compared to Freud's complete adjuvant, but it still serves the purpose. When you mix it, the milky suspension that you get in the emulsion form will suffice to induce immune response. Okay. In addition to these two types of adjuvants, what are the other adjuvants that are used, which are available in the market? They are the aluminum compounds, aluminum phosphates, or aluminum potassium sulfate, which is called as alum, or aluminum hydroxide. These are used as inert particles. And don't be under the impression that aluminum is toxic. It is not going to be administered in grams level. It will be in milligrams level. The particles are insoluble in aqueous phase. Since they are insoluble in aqueous phase, those particles can be coated because they are charged. They can be coated with the antigen of your choice. The antigen gets coated over them, and then you administer this particulate adjuvant to the individual once again through subcutaneous or intradermal or intramuscular route. And this vaccine is also not administered intranasal or oral or intravenous. Okay, these adjuvants, when they are used, they have to be administered only through the routes that I mentioned. So the most important vaccine that is administered like this with alum as a inert particle carrier is the one that is used to you as tetanus toxoid. Whenever you get injured, the vaccine that is available to you in the market as a booster is tetanus toxoid vaccine. They give you that shot of tetanus toxoid, which is coated over aluminum hydroxide. It also gives a light milky suspension because aluminum compounds are not soluble in water. So this way, the adjuvants are used to make the antigen available to the immune system in a very slow manner, very slow manner. So polio vaccine that is administered to you orally does not contain adjuvant. Probably the coronavirus vaccine that is going to be developed by the Bharat Biotech, which they are saying that they are going to give it as intranasal vaccine, may not contain adjuvant. But any other vaccine that is administered to you, intramuscular, subcutaneous, or intradermal, it will be definitely containing the adjuvant. And what is the properties of the adjuvant? How does it facilitate the boosting of immune response? The first effect is called as depot effect or depot effect. T can be silent. It acts like a depot. Depot means a storage region. Now, what is this storage region? Wherever you are injected, that site becomes highly inflammatory. And some of you may be still having that mark on your left arm, upper arm, a small blister-like mark there. And that was because of the BCG vaccination that was given to you when you were a child. But nowadays, for cosmetic reasons, for the girl children, it is not given on on the upper arm because it looks obvious. So for that reason, for girl children, they administer that vaccine in the foot pad. On the lower part of the foot pad, they will administer so that it will not be visible. But whatever may be the case, wherever you inject that vaccine, Bacillus tuberculosis vaccine, that is BCG vaccine, it will produce a blister, and that blister opens out and oozes for quite some time, and it is painful for the children at that age. So, whatever may be the case, the inflammation caused due to the depot effect causes the immune cells like dendritic cells in that area as well as the macrophages in that area to flood that area to sample the antigen. And that antigen which is distributed in that area will be there in the intramuscular or subcutaneous or dermal region. So the antigen presenting cells will sample that antigen and start processing and immediately migrate to the immediate nearby lymph node to present it to the T cells and B cells. And that is what is called as the second process, that is macrophage activation. And that blister formation is because of the granuloma formed by the macrophage. Granuloma is nothing but large assembly of macrophage in one place to tackle the antigen is called as granuloma. And those macrophages will remain there for quite some time in a large aggregation to form a granular appearance or granuloma-like appearance. Therefore, it gets the name granuloma. And they start secreting because they are activated now, because they have sampled the antigen, they will start producing the inflammatory cytokines. In addition, they will start secreting the cytokines that are responsible for the activation of the T cells as well as the B cells to tell them that there is an antigen that has come into the body and it is in large quantity deposited in one area. So these macrophages serve that purpose as antigen presenting cells. And that local inflammation caused by the infiltration of the macrophages there, plus the neutrophils also will come. Neutrophils will be invited to that area and they will infiltrate that area because in the inflammation, first cells to arrive are the neutrophils for phagocytosis. They will phagocytize these the particles, that is your vaccine, which is suspended in the oily emulsion, but they will not be able to present the antigen. But they will be responsible for secreting some cytokines also, and they are responsible for phagocytosis of that large mass of antigen and clear it. Okay, and that inflammation will be there for quite some time, about 15-20 days, and that pain will be there, and finally it subsides. Okay, ultimately, what is the purpose of adjuvant is to, in fact, activate T cells, and T cells, when they are activated, it leads to cell mediated immune response, and B cells, when they are activated, it leads to humoral, that is antibody production response. So that is the purpose of adjuvant, and that is the purpose which serves for boosting the immune response. So here the topic on antigens is over. Okay, we will move on to the next topic, and that next topic will be, in fact, the uh, topic on major histocompatibility complex. The major histocompatibility complex is nothing but the antigens that are, in fact, present on our cells, the nucleated cells, as tissue markers, which give identity to us and which gives a fingerprint identification, and it can tell which tissue the antigen belongs. In addition, the major histocompatibility, you have class 2 MHC cluster, which belongs to the complex with three different gene complexes, that is BP, DR, or BCA. Once again, when we are talking about autoimmune response, very often we'll keep mentioning these DP, DQ, DR for class 2 or BCA for class 1. So class 2, three different genes. Class 1, three different genes. They are nothing but their alleles. In terms, they are clusters of alleles. Therefore, there are multiple alleles. Then class 3 is made up of C4, C2, and BF. BF stands for the tumor necrosis factor. Okay, it is for the tumor necrosis factor. C4 and C2 belongs to the complements. C4 complement and C2 complement. They are produced from the gene cluster in the HLA complex, belongs to class 3. Okay, so as far as the genes are concerned, there are three major gene clusters: class 1 MHC cluster made up of BCA, and class 2 made up of DP, DQ, DR, and class 3 cluster made up of C4, C2, and BF. BF for the tumor necrosis factor alpha and beta. Remember, all these proteins that are produced from these genes are the proteins which form conjugate to form the glycoproteins. Okay, and class 1 MHC is a very important antigen by itself of a person, and that glycoprotein is expressed on all the nucleated cells. Please underline that statement. Any nucleated cell that is there in our body, it will express class 1 MHC. Okay, class 1 MHC is expressed by all the nucleated cells. Fine. And it is the characteristic of almost all nucleated cells that are present in our tissues. And it is a very important antigen. But there are some exceptions. Class 1 MHC is not expressed in our hair follicle. Okay. And class 1 MHC is not expressed in our cornea. Eye cornea. Cornea is the transparent region of the eye. It is not expressed there. Hair follicle and cornea do not express class 1 MHC. Therefore, since they don't express class 1 MHC, these two can be easily transplanted from one person to another person without rejection. They do not stimulate the immune response. Therefore, corneal graft from one person to another person can be easily done because it does not express class 1 MHC. Okay, so it is privileged tissue. Immune privileged tissue means it doesn't get rejected. It has a privilege of acceptance. Hair follicle can be transplanted from one person to another person without rejection. Okay, so that is one beauty of the two different um, tissues of our body. Okay, so let us see the structure of, oh, sorry, before that, see the distribution of class 1 and class 2 MHC on a particular cell. Okay, on a cell, class 1 is expressed, okay, and it is a nucleated cell. But if the class 2 is expressed, it is a nucleated cell, but it is an antigen presenting cell. It is only the antigen presenting cell have the special ability of expression of class 1 as well as class 2. Okay, class 1 as class 2, if both these class of MHCs are expressed on a particular cell, that means it is an antigen presenting cell. No other, please underline this point. It is a very important point. All nucleated cells do not express class 2 MHC, but all nucleated cells express class 1 MHC, which is a must, it is a tissue marker. But antigen presenting cells have a special ability of expression of both class 1 as well as class 2. That is the special ability. And they are nucleated cells. Macrophages and dendritic cells, being professional antigen presenting cells, have this ability of expressing both class 1 and class 2. Similarly, B lymphocyte, which is a non-professional antigen presenting cell, also expresses both class 1 and class 2 MHC if it comes in contact with the foreign antigen. Otherwise, it doesn't express class 2 MHC, but it will express class 1 MHC. Now let us see the characteristic features of class 1 MHC. The class 1 MHC molecule is a glycoprotein made up of an alpha chain, and it is in fact present as a self antigen on all nucleated cells. The alpha chain is divided into three domains: alpha 1 domain, alpha 2 domain, and alpha 3 domain. And then that alpha chain is inserted through the membrane. Okay, that is, only alpha chain is inserted through the membrane, and that region where it is there in the membrane is called as transmembrane segment or transmembrane domain. And there is a cytoplasmic tail which is hanging into the cytoplasm. Okay, and there is another molecule which is associated with it, that is beta 2 microglobulin. Okay, it is present as beta 2 microglobulin, which associates, and it is produced separately by another gene. It is not produced by the MSC complex gene, it is produced by some other gene, and it comes and associates with the alpha 3 domain of the class 1 MHC to constitute the molecule as a whole molecule. But remember, beta 2 domain, beta 2 microglobulin doesn't have contact with the membrane. It is just having an established contact with alpha 3 domain. Okay, and class 1 MHC molecule is capable of expressing all self antigens of the endogenous origin. Those antigens that originate within the cell are presented on class 1 MHC molecule in this region, that is the region called as peptide binding cleft. In the region where on the top, where alpha 1 and alpha 2 domains form a platform, there is a cleft or a groove, that is antigen binding groove. There the self antigens are presented. The self antigen belonging to a cell is presented there. Now occasionally the class 1 MHC will also present the antigens of pathogenic origin if the pathogen is there within that cell. At the same time, if a cell has transformed into a cancer cell, that cancer antigen is also presented in the peptide binding cleft or antigen presenting cleft. So therefore, the self antigens plus the antigens that originate within the cell are presented on class 1 MHC. When it is presented on class 1 MHC, it is going to be recognized by the T cytotoxic cells. Please remember this. It is a thumb rule. Any antigen that is presented on class 1 MHC is going to be recognized by the CD8 T cell, or it is called as TC cell or T cytotoxic cell. Once again, it is responsible for presenting the self antigens, but if it presents non-self antigen, T cytotoxic cells will be activated. Then NK cells also are capable of recognizing the antigen presented on class 1 MHC. Okay, and mentioned that the class 1 MHC is produced by A, B, C gene cluster or alleles. There are 59 A alleles, 111 B alleles, and 37 C alleles in that gene cluster. And that allele expression, which is multiple allelic expression, goes in permutation and combination, and therefore in any individual, healthy individual, six haplotypes like this. See, there are 6 haplotypes. On top here, the class 1 is expressed on a cell. 6 haplotypes will be expressed at any given time. And all these six haplotypes are the tissue markers of that person. Okay. Then the class 1 MHC, in fact, has other characteristics. What are the other characteristics? You know that it is made up of 2 different proteins, alpha and beta, and alpha chain and beta chain associating. Therefore, it is called as a heterodimer glycoprotein. It is a glycosylated protein. So the alpha unit of the class 1 MHC is about 45 kilodaltons in molecular weight, and beta microglobulin, which comes and joins it separately, is about 12 kilodalton in molecular weight. Okay, and each domain of the alpha, that is alpha 1, alpha 2, and alpha 3, they have intra-chain bonds made up of disulfide to give foldings in the protein. Okay, in the protein, and that intra-chain bonds gives compactness, and there are about 90 amino acid residues in each of these domains. That is alpha 1, alpha 2, alpha 3 have 90 amino acid residues, and the transmembrane domain, this which is there in the membrane region of the cell, is about 40 amino acid residues, and the cytoplasmic domain is 30 amino acid residues, and that is responsible for the signal transduction. And by having this constitution, the class 1 MHC molecule is responsible for presentation of the self antigens. And why does that beta 2 microglobulin, which is produced separately, comes and associates with the alpha 3 domain, is because of the homology of the amino acid sequence of the alpha 3 domain and beta domain. And it is observed that that homology sequence of the amino acids here resemble the constant region of the antibody. Therefore, the class 1 MHC molecule or class 2 MHC molecule, which we will come across later, are called as immunoglobulin superfamily. Whenever they have some resemblance to the immunoglobulin domains, they belong to the family of immunoglobulin superfamily. Okay, and it is observed that alpha 3 domain is highly conserved, and it may be common in many of the class 1 MHC molecules that are expressed by different individuals. The diversity is there in the alpha 2 and alpha 1 domain, but alpha 3 domain has conserved amino acids residues, and it plays a major role in the sense that the CD8 receptor of the TC cell, CD8 receptor of the TC cell binds to this domain, alpha 3 domain, when class 1 MHC is going to present the antigen in the antigen binding cleft. When the antigen binding cleft is engaged with the antigen, the TC cell, which recognizes that when it forms a synapse with it, the CD8 receptor of the TC cell forms a contact with alpha 3 domain, and it is a must. Without that, CD8 cell cannot be activated. Okay, so the peptide binding cleft is made up of the alpha 1 and alpha 2 domains as platform, and at a time, to the cleft, the amino acids, that is the peptides residues, the peptides that can bind are not more than 10 amino acid residues. The peptide chain will be of 10 amino acid residues, not more than that. It can be anywhere between 8 to 10. And generally, the peptide that binds there in the antigen binding cleft is called as a nonamer, because predominantly 9 amino acids will be there, but it can go up to 10 amino acids. And all those peptides that bind the cleft, antigen binding cleft, have some common features, means they have some common amino acids. They may be different peptides, but by thumb rule, they must have a common residue of amino acid at certain positions to interact with the class 2 MHC. And the common amino acid is highly conserved among the different peptides that are generated. And those conserved residues are called as anchor residues. Anchoring means for lodging. And without that anchor residues, the peptide cannot bind to the antigen binding cleft. And it is observed that those anchor residues are predominantly in the class 1 MHC that binds there with the peptides is leucine and isoleucine. The residue 2, that is second amino acid, is always leucine and or isoleucine. The ninth amino acid is always leucine and isoleucine. So, in between amino acids, that is the first and then the remaining amino acids, could be different, but these two amino acids, that is in the second amino acid and the ninth amino acid, cannot be different. They're conserved and they have to be leucine and isoleucine in all the different types of peptides that are generated endogenously. They bind to the cleft of the class 1 MSC. If they don't contain these amino acids, it cannot bind them. The bound peptide, that is, which is bound to the class 1 MSC, generally arches away, and it arches away in this manner. Okay, like it is in a convex manner. So the second amino acid lodges, anchors, ninth amino acid lodges. In between amino acids do not associate, and it will be a convex appearance of the antigen peptide. And that is what it makes an efficient peptide presenting molecule. So this class 1 MHC is easily recognized by the TC cell. Any strange peptide that is going to be presented here will make it a foreign peptide. It could be of intracellular pathogenic origin, or it could be the strange antigen that is produced by the cancer cell that stimulates the TC cell to carry out its activity of cytotoxicity or killing. Okay. Then coming to class 2 MHC. The class 2 MHC molecule is also a heterodimer. It is made up of two different chains compared to the class 1 MHC. The two chains which are heterodimers, alpha 1 and alpha 2, beta 1 and beta 2. Both of them are embedded in the membrane. Whereas in class 1 MHC, beta 2 is free. It is not embedded in the membrane. Please remember that. Do not make a mistake of connecting it to the membrane. It is associated with the alpha 3 domain, but not connected with the membrane. Whereas here in the class 2 MHC, the beta 1 and beta 2 domains, which form a separate peptide that traverses through the membrane and goes into the cytoplasm as a cytoplasmic tail. And I already mentioned to you that class 2 MHC is expressed by only antigen presenting cells. Okay, so even though the two chains, alpha and beta chains, are traversing across each other, they do not have any covalent association with each other. They are just folded within themselves with disulfide bonds within themselves, but no disulfide bond between alpha and beta chain. They are free from each other, but they are associated in such a way that the folds will move into each other of the alpha 1 and beta 1, and they will be traversing across to establish the contact. Okay, so there are, like in class 1 MHC, there are 6 haplotypes expressed on a cell. Here, 12 haplotypes of class 2 MHCs are expressed on an antigen presenting cell. It is 2 different peptides, therefore it is a heterodimer. And alpha dimer, alpha chain is about 33 kilodaltons and beta is about 28 kilodaltons. Okay, and the transmembrane domain, once again, it is about 40 amino acids and 30 amino acids of the cytoplasmic region. Okay, so alpha and beta domains do not have any covalent interaction, but they are in fact interacting at a region where alpha 1 and beta 1 will have traversing chain within themselves. Okay, and alpha 1 and beta 1 domain on top forms the platform for the antigen binding cleft. Whereas in the case of class 1, it was the alpha 1 and alpha 2, but in the class 2, it is alpha 1 and beta 1. And the peptide presented on the cleft of the class 2 is always an exogenous antigen. Exogenous means it doesn't belong to your cells. It is of the bacterial or viral origin or coming from outside and processed by the antigen presenting cells and presented there in the peptide binding cleft. And what is the specialty? These peptides are of about 13 to 18 amino acid residues, larger peptides compared to the peptides that are bound in class 1 MSC. And of the 13 to 18 amino acid residues, they have anchor residues made up of, the proline or hydroxyproline residues. Okay, and these residues are present in the second position and in between and the terminal region, second position of the amino terminus, in between and carboxy terminus, and the proline residue which is present, which is capable of anchoring, it becomes an anchor residue, and the association of the peptide will be like this. And there will be extension of the peptide outside the class 2 MHC, which was not seen in class 1 MHC. Class 1 MHC, the association was very, very clean, and there is no extension outside. But whereas in the class 2 MHC, the interaction is haphazard, and the interaction will be in the second, then in between, anywhere between 7th to 10th amino acids, and then the carboxy terminal region. Okay, the proline residues. So the peptide could be of 13 to 18 amino acid residues, and it is generated from the extracellular or extraneous pathogen of bacterial origin, and that is responsible for presentation to T cells, T helper cells. The T helper cells will be responsible for receiving the antigen presented by class 2 MHC. I'll stop here. Thank you.